Hello and welcome to another neuroanatomy video. I am Tanya Chamberlain from the Division of Anatomy here at the University of Leeds, and in this video we will be tuning in on the auditory system. Before we go any further, let's have a look at today's learning objectives. After this video and any necessary further study, you should be able to identify the parts of the auditory pathway from the outer ear to the cochlea. Outline how sound waves are converted to an electrical impulse. Outline the pathway of the auditory system from the cochlear nerve to Heschel's gyrus, the primary auditory cortex, and describe the tonotopic organisation of sound and frequencies on Heschel's gyrus. First, let's focus on the components of the auditory pathway. Here we have a profile or lateral perspective of the head and face. On this image we can see the projection of the skull with the individual bones outlined. The bone we are interested in is the temporal bone. In the temporal bone we can see the opening of the ear or the external acoustic meatus. This image is a superior view of the cranial base. What we have done to get this image is to remove the calvaria or skull cap, and we have pulled the brain out and all the other soft tissues, so we are looking down into where the brain would sit. Here we can identify the temporal bone. I have not coloured the right one in, but you can see where it is. We can see something called a vestibular labyrinth, which sits within the bony part of the temporal bone, called the petrous part. Petrous literally translates to rocky or hard part, which is how we can remember the characteristics of this region of the temporal bone. Let's go and have a look what it looks like from a different perspective. On this image we can see how the vestibular labyrinth sits within the petrous part of the temporal bone. It was superimposed on the last picture, which made it a little difficult to visualise. On this coronal section we can see the course in which sound waves are going to have to travel to reach the organs that will relay the information to the brain. Let's talk about the different regions of the ear first. The first part is the outermost part, or the external ear. It is composed of the auricle, which is the visible funnel that is guiding sound waves into our external acoustic meatus. If you stick your finger in your ear, which I 100% do not advertise you do, you are sticking your finger in the external ear. The inner boundary is formed by the tympanic membrane, commonly referred to as the eardrum. The middle ear is the next stop. This is an air-filled space where sound waves are converted into mechanical messages by three tiny bones in the ear called the ossicles. The inner ear is formed from the vestibular labyrinth, which converts the mechanical messages into electrochemical message, which is transmitted to the brain by one of the cranial nerves. Let's have a closer look at each region. So the external ear is composed of the cartilage auricle, which helps to funnel sound into the external auditory canal. The outer or lateral most third of this is constructed of cartilage, whilst the inner or medial 2/3 is constructed of a bony material from the temporal bone. When we look at the inside of the external auditory canal, we can see a thin membrane forming the wall that divides the external ear to the middle ear. This is the tympanic membrane. This should be translucent and intact, as indicated by the image on the right. As sound waves enter the external ear, the vibration of the air bounces on the tympanic membrane, which will cause it to vibrate in response. The tympanic membrane separates the external from the middle ear. The middle ear is an air-filled space. This part of the ear communicates with the pharynx via a eustachian tube, which helps to equalise the air pressure in the middle ear with the external environment. The middle ear contains tiny bones which relay vibrations from the tympanic membrane to a complex in the inner ear. The middle ear bones are the malleus, the incus, and the stapes, which articulate with each other. The malleus attaches to the tympanic membrane, and as the tympanic membrane vibrates, this movement is projected forwards via the incus to the stapes, where it bounces on a membrane called the oval window. On this penny on the top right, we can see how tiny these bones are. If you are ever asked in a pub quiz what the smallest bone in the body is, you will be able to confirm that it is in fact the stapes, or the stirrup bone of the middle ear. The eustachian tube plays an important role in balancing the pressure of the middle ear environment and the external environment. But, as it is a tube that stretches from the pharynx to the middle ear, it can be quite problematic for transmitting infection. This is why when we have an upper respiratory tract infection like a common cold, we can see them present as earache or ear infections. The eustachian tube in the adult is at an acute angle, meaning that mucus drains fairly effectively into the pharynx when swallowing, ensuring a quick evacuation of bugs that have found their way in. However, if we look at the child image on the left, we can see that they are far more horizontal, which means that drainage is not quite as efficient. This is why children are more prone to conditions like middle ear infections, or otitis media, or conditions such as glue ear. So now we are looking at the middle ear in the direction of the arrow at the left of the screen. I have peeled off the tympanic membrane so we can see inside the air-filled cavity. This is considered a high-risk space because we have a lot of structures passing in close proximity, and we know that this structure is going to be susceptible to infection because of the eustachian tube. Infection doesn't end there, because on the posterior wall we have tiny holes called mastoid air cells, which are able to transmit infection to the middle cranial fossa. Directly below the middle ear is the internal jugular vein, which can thrombose. Anterior to the middle ear is the internal carotid, which can induce a condition called pulsatile tinnitus. And finally, as we've seen in other modules, the chorda tympani and the facial canal transverses the middle ear. Thus, a middle ear infection can cause a lesion or swelling and disrupts the innervation of some of the facial muscles of expression. Now we should move on and look at the inner ear, which will form a great part of the remaining video. The inner ear is constructed of a bony vestibular labyrinth. This labyrinth is constructed of two regions, a vestibular region, which conveys information to the brain regarding balance and body orientation, and the cochlear part, which detects and transmits sound. The vestibular part is the region in which large loops like a roller coaster, whilst the part we are interested in is the one that looks like a snail shell, the cochlear part. The inner ear is located in the petrous part of the temporal bone, as indicated early on in the video. It, as mentioned, is composed of two special sensory organs. The vestibular system, which is responsible for balance and equilibrium, is made up of the vestibule and the semicircular ducts and canals. And it is made up of the cochlea and the cochlear ducts. Information is taken from these special sense organs and transmitted to the brain via the cranial nerve 8, the vestibulocochlear nerve. The vestibular part conducts information from the vestibular system, and the cochlear part conveys information from the cochlea. The inner ear inside the petrous temporal bone has a series of fluid-filled cavities. The outer circumference is bony, and the bony labyrinth contains a liquid called perilymph. We then have a membranous part, which contains a liquid called endolymph. We can remember this by peri meaning perimeter and endo, which means inside. So the perimeter is perilymph and the endolymph is the inside liquid. Perilymph is located in the bony parts, is similar to extracellular fluid, and endolymph is like the inner cellular fluid. So let's start building up what we have touched upon using the image on the bottom. Sound waves enter the external canal and cause tympanic membrane to bounce. This tympanic membrane transfers the sound waves to mechanical motion and passes them on through the 3 bones of the middle ear through movement. The final bone, the stapes, bounces this movement, similar to a high-tech Morse code, on the oval window, which separates the middle ear from the inner ear. Behind this window sits the perilymph, which is a very good conductor of sound. Let me draw this on for you. Now, each region of this membranous labyrinth is triggered by a particular frequency and or volume or amplitude of sound. High frequency sounds are picked up at the beginning of the spiral, here, whilst low frequency sounds are picked up from the apex of the snail shell, which would be here. Along the length of this, we have individual cochlear fibres that conduct sound back to the brain. Within the labyrinth, we have 2 chambers and a duct that separates them. We have the scala vestibuli and the scala tympani. And the duct that separates them is called the cochlear duct. These chambers are continuous with each other through a narrow slit at the apex of the cochlea, or the snail shell part, through a helicotremor, which allows endolymph to move through the chambers and therefore sound waves to travel. Let's have a closer look at the scala vestibuli, the tympani and the cochlear duct. Above the cochlear duct lies the scala vestibuli, and this separates this from the cochlear duct. And the separation between this and the cochlear duct is the vestibular membrane. Below the cochlear duct is the scala tympani. The floor of the cochlear duct is constructed of a basilar membrane, which contains a very special region called the spiral organ of Corti. Each of the chambers and the cochlear duct there is endolymph which helps to conduct sound. We can also see that one of the fibres of the vestibulocochlear nerve travels to the spiral organ of Corti. The spiral organ of Corti requires some special attention now. The spiral organ of Corti is moved by deformation of the cochlear duct. The cochlear duct is moved by perilymph of the bony labyrinth carrying vibrations from the transmission of sound. As the cochlear duct moves, the spiral organ of Corti moves. When this happens, the hair cells from the spiral organ of Corti are embedded into the tectorial membrane, which causes a transmission of signals from the tectorial membrane to the hair cells. This triggers an electrochemical transmission to the fibres of the cochlear nerve, which are conveyed to the brain. Here is a summary slide. I will not read this to you, but pause the video if you wish to recap this information. So now we have covered the first part of the video, let's have a look at our next learning objective and see how these messages are going to get back to the brain. Here is an overview of the auditory pathway which we will cover today. The auditory pathway is polysynaptic and topographically organised. We can see this on the image. Looking at the cochlea, the low frequency sounds are transmitted from the apex of the cochlea, whilst the high frequency sounds are transmitted from the beginning of the spiral. We know these regions have cochlear fibres, so we know that these fibres synapse and eventually make their way to the thalamus. The thalamus will distribute these to different regions of the temporal cortex, and we will look at this shortly. Additionally, the organisation of the pathways is bilateral, so sounds entering the right ear will be transmitted to both sides of the temporal cortex, markedly different from what we saw with the eyes and other pathways we have covered. It is used to compare the timing and the loudness, which will help us identify where this sound has come from. I'm sure we're all familiar with cranial nerves now, so let's quickly glance at the vestibulocochlear nerve, arising from the pons. This is where messages are entering the brainstem. In order to look where the cochlear fibres synapse, we need to flick the brainstem around to a dorsal perspective. The cochlear nuclei span the junction between the pons and the medulla. Above this, we have the superior olivary nuclei and the inferior colliculi. And lastly, we have the medial geniculate nuclei of the thalamus. All of these centres are the auditory centres, where we are going to expect auditory fibres to synapse, and potentially we will have some other systems synapse and influence the auditory system. Also, we have the inferior brachium, which is a tract in which some auditory fibres find themselves travelling between the inferior colliculus and the medial geniculate nucleus. So let's have a look at what happens. The first order fibres begin at the hair cells and enter the brainstem at the cerebello-pontine angle, where they synapse with the second order neurons in the dorsal and ventral cochlear nuclei. Look at the sketch. Here we can see the first order neuron at the cochlear nerve. These synapse at the dorsal and ventral nuclei. This image requires a little orientation, as the image on the left is taken from the picture of the brainstem on the right at the level indicated. But the brainstem is from a dorsal perspective, and the sketch on the left shows the ventral at the bottom and the dorsal at the top. If I add in the fourth ventricle, this should help to keep you oriented. Let's focus on a schematic. The pathway is as follows. The cochlear fibres, with their spiral ganglion, transmit the first order neurons to the cochlear nucleus. Here they synapse onto second order neurons. From this, the second order neuron is going to enter a region called the trapezoid body. Here, most of the fibres will decussate or cross over to the other side before ascending. Not all of the fibres cross. Some will remain on the ipsilateral side to the first order neurons. These fibres then synapse in the superior olivary nuclei and enter the lateral lemniscus tract. As they travel up to the caudal midbrain, more specifically to the inferior colliculus, the fibre enters, they synapse again. From the inferior colliculus, they ascend further to the medial geniculate nucleus of the thalamus via a tract called the inferior brachium. After synapsing at the medial geniculate nucleus, they then travel to Heschel's gyrus. Now please be aware, should you consult other texts, this is an incredibly slimmed down version of all of the pathways that can take place. Variations in the pathways begin as far back as the synapsing of the first order neurons in the cochlear nuclei. So depending what level of neuroanatomy text you find, you will find various pathways for the auditory system depending on the neuron and the stimulus type. Now cast your mind back to the second learning objective, where we discussed the regions of the cochlea. Different regions of the cochlea detect different frequencies or pitches or vibrations produced from the sound waves. This is called tonotopic organisation. The apex responds to low pitch, whilst the base responds to high pitch. The frequency is distributed to different regions of the cortex. Low frequency from the apex is taken to the anterolateral part of the Heschel's gyrus, whilst high frequency from the base is transmitted to the posteromedial parts of the gyrus. You could think of low sound being transmitted from the anterolateral part to help you remember. As discussed right back at the beginning of this module, each hemisphere is dominant in specific roles for most people. In most scenarios, the left hemisphere is dominant for language production and comprehension. We know that we have two secondary auditory areas of interest. We have Broca's area, located in the frontal lobe, and we have Wernicke's area, located on the temporal and stretching into the parietal lobe. We can consider two forms of aphasia. We have Broca's aphasia and Wernicke's aphasia, which are due to damage to their respective areas. The way that I remember them is by thinking Broca's deals with broken speech and Wernicke's deals with weird words. Let's have a look at a couple of examples. Case 1 and Case 2 were asked to describe what they saw in the picture. Case 1 said picnic, flying kite, swimming, house, tree, pouring water, boats, trees, okay, car. Pause the video and think what area is affected and what mechanism of damage to this area might have it arisen from. If you said Broca's area, well done. You identified the broken speech. This could arise from an infarction to Broca's area from an anterior branch of the left middle cerebral artery. Let's study Case 2 now. In the pier, somebody is fishing, somebody is playing into the water. The man with the kite has the jeans with the pocket deal. Pause the video and see if you can identify where this aphasia arises. This is Wernicke's area that results from a posterior middle cerebral occlusion. Let's briefly have a look at these arteries. The anterior distribution of the middle cerebral artery travels to and supplies Broca's area, whilst the posterior distribution supplies Wernicke's area. There is one final thing to think about before we can draw an end to this video. Let's look at the auditory reflex. In response to sudden loud sounds, we can become startled. When startled by a loud noise, the superior olivary nucleus immediately sends out messages via the facial nerve and the mandibular branch of the trigeminal nerve to contract the muscles of the ossicles in the ear and dampen the sounds to protect the hearing. Occasionally, the inferior colliculus sends messages to the eye muscles via the cranial nerves 3, 4 and 6 and to the spinal nerves in response to sudden sound. This is to get our eyes and head face the direction. Occasionally, this may also induce a jump-like startle if there was enough stimulus from the auditory pathway. And that brings us to the end of this video. You should now be able to identify the parts of the auditory pathway from the outer ear to the cochlea. Identify how sounds are converted to an electrical impulse. Outline the pathway of the auditory system from the cochlear nerve to Heschel's gyrus in the primary auditory cortex. and describe the tonotopic organisation of sound frequencies on Heschel's gyrus.